Good evening, ma'am. Good evening, ma'am, sir.
Ma'am, good evening. Okay, we'll start the class. In the previous class, we have done some of the important concepts. In this class also, we will be dealing with the concepts only. Each and every concept is a class. You remember, I am trying to compress all the concepts in few only in two classes. Last class I have done some of them, and now I am going to complete or close it. Okay. Why is immunology important? Can anyone elaborate how it is useful to you in your career or in holistic? Can anyone?
Sir, may
Please go ahead.
Yes, sir. Sir, we have been seeing so many autoimmune conditions, right, sir? I mean, it's an altered way of immune response only. Even in case of, a normal immune response also, like it could be as simple as viral infection or bacterial infection. It's better we know how our body works, because we also looking at the blood reports and everything, like C-reactive protein, why ESR is elevated or something. So it is giving us a fair idea like what is happening inside the body, and thereby we can also having the holistic understanding. I think having this understanding is also very much necessary for us. Together combined, I think our approach could become much more better.
Perfect. Okay. So okay, let's move ahead. So in this slide, we are comprehending what we have learned till now. The microbe is entering inside, that is when the microbe is or the pathogen or an antigen, different names. Okay, microbes, pathogens, and what is the antigen? I will elaborate you. It's entering, it's breaking the barriers. So once it is entering, the innate immunity is getting activated in the form of complement or resident cells like dendritic cells or macrophages. And once they get ingest these antigens, they activate different chemical different chemicals are released. They're called as cytokines altogether, and also chemokines. Cytokines and chemokines are the two different chemical molecules that are being released during the process of immune activation. So in this, there are interleukins. They stimulate the natural killer cells, the liver, and the liver releasing the acute phase proteins. And you have mentioned the C-reactive protein. That C-reactive protein or the CRP that is being released by the liver. Okay, so that's why we measure CRP in an indirect way to estimate the quantum of inflammation or the interaction between the pathogens and the host. That is an indirect way of measuring CRP. Mainly, the CRP is measured in pediatrics. In adults, CRP is not that much reliable, but okay, to a certain extent, we can rely on it. So it's not 100% the molecule to measure. We also measure procalcitonin and all those is acute phase proteins. Okay, that is a different class. Okay, if sometime someday if we get a chance to read about these acute phase proteins, we'll deal it. Okay, so that's causing the fever, and the fever is the symptom that we see because of the inflammation, because of the antigen and the immune host immune mechanisms interaction. So causing the vasodilatation, that means increasing the size of the blood vessels. Means a lot of blood is being pushed there at the site of activity, and from there the various cells they squeeze out of these blood vessels, and they reach the target area through the process of the chemical gradient that has been established from the site of infection to the blood vessels. Okay, and then we see the signs of inflammation around the area of the site of infection. Okay, I told about the complement, which is also an innate immune mechanism. It is also activated, and the end result is the microbe is directly destroyed, or it is opsonized, or leukocytes are being activated to the site of infection because of the complement molecules that are present inside the blood. So complement is present inside the blood. Okay, so this is the story so far. Okay, and now we move on to the main molecules, the heroes of immunology. One is the antigen, and another one is an antibody. Okay, antigen. What is an antigen? Antigen is if say a microbe is entered, say some typhoid bacilli, salmonella typhi, its name. So on its surface, there are different molecular patterns or structures that are present on the surface of the salmonella. That is, you see the Vidal test. Vidal, if you go for a biological test, for a blood test, they will do Vidal for typhoid. They measure the O antigen and the H antigen. So O is the somatic, that is the somatic antigen. H is the flagellar antigen. Flagella is the surface structure of the bacteria. Flagella, with the help of flagella, the bacteria moves. So it is the locomotory organelle of the bacteria. So that flagella contains the antigen H. Similarly, the cell wall contains O antigen, that is somatic antigen. So we are measuring, we are estimating the O antigen load and flagellar antigen is the H, Vidal O and H. We give that some value, okay, that will be knowing in the next few coming slides. So that is the antigenic part. What is antigen? Antigen is the smallest molecular structure that can be able to produce the immune response, which can activate the immune molecules. That is called as an antigen. So from now onwards, we'll be talking only antigen, not that now the parasites, bacteria, viruses, we are not going to talk antigen. So bacteria contains antigen, parasites contains antigens, viruses contains antigens. The smallest molecule. So we are now entering into the molecular level. Okay, so it's like Newtonian science and Einstein or quantum science. Okay, the laws, basic laws, and now we are moving on to advanced laws. So here we talk about only the antigen and antibodies. Okay, the antigen may be present across all the parasites which are entering, means something that is causing the disease. Okay, in this slide, this is the structure of the antibody. What is this antibody? The antibody is a molecule that is produced in the in our immune by our immune cells. I will tell which cell produces these antibodies by our immune cells, which go and fight or bind to the antigen, and in turn it gets either neutralize or destroy or will do something else that is helpful to us. Okay, is it clear? Is it clear? Are you able
Yes,
to
sir.
understand till now? Right, very good. So you remember, so that is the antigen and this is an antibody. And which cells produce antibody? B cells. You know what are B cells? B cells. What are these B cells? What type of cells are B cells? Can anyone tell? Last class I have said B cells. What type of cells? They are lymphocytes. Okay? Natural killer cells, T lymphocytes, B lymphocytes. Okay, these all come under the lymphocytes. Is it clear? Among the lymphocytes, B lymphocytes will produce the antibodies, right? So these antibodies are produced by B lymphocytes. Okay, this is the structure of uh, uh, the antibody. Okay, so it is like a Y shape. Y shape. Its structure is Y shape. That's you can see in the structure that is the epitope binding region or NH terminals, and there is a one red color round structure that is antigen. Okay, so two forks are coming. So those forks, they will go and bind to the antigen. Okay, and that blue color arm, that's extended part, is a heavy chain. Okay, that is the uh, CH4 terminals, COH terminal we called. That will go on, that will be either free or it will bind to the cell. Okay, and those forks which are present in the Y shape, that will bind to the antigen. That is an antigen binding site. Is it clear? I am telling very simply. I am not at all going to detailed molecular structure or other thing.
Yes,
Only
sir.
for your understanding purpose, I am trimming down all the other concepts. Only I am trying to introduce the important point into your brain. Okay. So this is the molecule that will go and bind to the antigen. This is an antibody. It is Y in shape. Okay, it having two light chains and heavy chains. Okay, so this is the antibody. And this slide, you will see there are different shapes. Okay, different shapes are different formations of these antibodies. There is a single molecule, the first one, and then there are 5 molecules. 1, 2, 3, 4, 5. They all join together, 5 molecules. Okay, and if you go down, there are 2 molecules joined together by J chain. Okay, so these are the different patterns of these antibodies. Okay, and basing upon this, the composition of light chain and heavy chain, they have been divided into 5 classes. Immunoglobulin, GAMED. GAMED. Immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin E, and immunoglobulin D. These antibodies are also called as immunoglobulins. Did you get the point?
Yes, sir.
Are you
Yes,
with me?
yes, sir.
Are you with
Uh,
me?
yes,
Okay,
sir. Yes.
because
Yes, sir.
I have received a complaint in, from Suresh sir that in the previous class that I am going my way, but how far your students have understood, you please consider about it. That's why I'm okay. I'm trimming down the maximum data so that the basic points will enter. Okay, so there are 5 classes of immunoglobulins or antibody. Now you forget antibody. Now, okay. Now onwards, I call them as immunoglobulins. There are 5 types of immunoglobulins. There are, again, I'm repeating, GAME-D. The mnemonic is GAME-D. GAME, GAME-D. Immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin E, immunoglobulin D. G for gamma, A for alpha, M for mu, D for delta, E for epsilon. Is it clear? Okay,
Yes.
5 types of immunoglobulins. GAME-D. Okay, GAME-D. Immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin E, D. So now you forget immunoglobulin. The next now I am introducing IG. Immunoglobulin short form is IG. Okay, game deal. IgG, IgA, IgM, IgE, IgD. Is it clear?
Yes, sir.
Yes, sir.
Immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin E, immunoglobulin D. IgA, IgM. So you see, for example, dengue test. Dengue, all of you have seen, I think, dengue test. So in the dengue test, they will give IgM, IgG. Have you noted it? Have anyone have seen that
Yes, sir.
test report? They will give IgM positive, IgG positive. What is that interpretation? It's interpret that immunoglobulin G against the dengue antigen is positive. Is it clear? Immunoglobulin G against that dengue antigen. Okay, that is IgG is positive or IgM is positive. Okay, and in these 5 classes, they are present in different regions. Okay, they are present inside the blood. They are produced by the B cells. the B cells, they produce the antibodies, and these are 5 classes of antibodies that are present. So, Emma, I will tell now how they look like. IgG, okay, IgG is a single molecule, IgG. Okay, IgM is a these 5 molecules are there now in this slide. If you see, there are 5 antibody molecules joined together. It is an IgM structure. Its molecular weight is also high. It's five molecules joined together. That is immunoglobulin M. Did you get my point?
Yes, sir.
Yes or no?
Yes, sir. Yes, sir.
IgM. Okay. I am going very slow. Why? Because this is very, very critical for understanding the various serological tests that we interpret in the lab data. Okay. So this is IgM molecule. And then IgA, it is a dimer. That means 2 molecules joined together. Immunoglobulin A, IgA, is a dimer. Okay. IgE, IgG, IgD, there are monomers, means single molecules, but with different molecular weights. So you don't bother about the molecular data and everything. You just don't bother. You just try to understand how their shape look like, what is their valency. What is meant by valency? Valency is how many antigen molecules a antibody can bind. That is the valency. A monomer means a single molecule can bind to 2 antigens with its with 2 arms. Is it clear? Is it clear?
Yes, sir.
So one antibody molecule will bind to 2 antigens. Then if you go here, IgM molecule will bind to how many antigens? Give the answer.
10, sir.
How many?
10.
10. Very good. IgA?
IgA 2, sir.
4. 4. 4. IgA
Four.
is
Very
2
good.
dimer.
It's a
4.
dimer.
Very good.
Yeah.
Yes. You are all
Okay.
are with me. That I can sense. Okay. So now among these molecules, which is powerful, do you think?
IgM, sir.
Why?
Because it has 5 molecules. I think Ig.
You don't think
Uh,
you don't go further. You stop there only. Hmm.
okay, sir.
Hmm.
IgM,
So
sir, because 5 molecules and 10 antigens.
so it is neutralizing 10 antigens. So 10 valence. It is fighting with 10 valence. Okay. So that's why you think it is powerful, right?
Yes, sir.
Yes. So yes, exactly. IgM is very useful to us. Okay. Why? Because it is able to bind 10 antigens. That's why the important point you have to get here is early in the infection, very early in the infection, the antibody that is produced is, what is the answer?
IgM, sir.
Very good. Excellent. So early in the infection, the antibody that is produced is IgM. So don't worry about this. Why I am sliding here, because I want to. Oh yeah. So right. This slide. Okay. So in this slide, if you see, this slide interprets that means the um, the antigen and antibody interaction slide. Okay.
Okay.
So early in the infection, the antibody that is produced is IgM. Why? Because its valency is 10, right? So if the less valent molecule go and starts fight for us, will it be useful to us? Yes or no?
No, sir, because obviously these virus they multiply more.
Exactly.
Quickly.
So that's why now the funda is IgM appears early. Later on, IgG appears. You note it down. You note this point. It is applicable to all the infections, universal. The point that I mentioned is universal. It will not change. There are no exceptions. Early in the infection, IgM is produced. Later on, IgG is produced. So if a dengue test, IgM is positive, IgG is negative, what is your interpretation?
So
Body tried to fight.
recent occurring
Um,
of the infection.
Exactly. It is less than 3 days.
okay.
It is less than 3 days. IgM positive. If a test report gives IgG positive, what does it indicate? There is no IgM, only IgG is positive. What does it indicate?
It's been long time since the infection is
Exactly.
present.
It is a chronic infection. That's it. Okay. It is a chronic infection. It have occurred long back. Even it 1 month back, it may have occurred. Okay. It's IgG. Okay. If a test result both IgM positive and IgG positive, what does it indicate? Now this
So
is tricky, but you have to answer.
it has been there, but it
Hmm.
might be a very very
Hmm.
strong infection that has entered. So both of them are
Hmm.
simultaneously occurring, or there might
Exactly.
be two different
Hmm.
infections that occurred.
Exactly. If both IgG and IgM is positive, maybe there is a chronic, that is a previous antigen is there. It have been neutralized. But still its IgG molecule is present. Okay.
Yeah.
Now new antigen have entered, that is IgM. Is it clear? Is it clear now?
Yes.
Yes.
The interpretation of IgG is IgM. Okay.
Mm.
And now is IgG molecule is very specific molecule. It is very
Sir,
yes,
uh,
please.
sir, I have a small doubt before you
Yes,
go ahead with the
yes,
class. Sir, this
yes.
IgG molecule, you said it is a single molecule, and
Exactly.
IgD and E are monomers. I mean, G, D,
Hmm.
E, all are same, or it is different, sir,
They are
again?
different. Why I didn't call with same name? No.
No, sir. What I'm trying to understand here
Hmm.
is
Hmm.
here IgG single molecule, you said.
Yes.
That means it can catch up to 2 antigens, right?
You forget about how you forget about it's only
Valence.
valency. How many antigens it can
Mm-hmm.
catch is only valency. But
Okay, sir.
they are different. They are different
Okay, okay,
monomers, but they are different. We are all humans,
okay,
but not same.
okay, okay, sir.
They
Some
differ
of
in
them
their
might be
molecular.
impacting the different antigens.
Yeah, they
Yeah,
differ.
I
Yeah.
understood, ma'am.
They,
But the protein chain or something that I'm trying to understand.
that's why it is a big class. Immunoglobulins is a big class. Okay.
Okay, sir.
But I don't have liberty to explain all these things. Okay.
Okay, yeah.
Because you should get it in compact way, but in a useful way. That is our primary motive for taking these classes, right?
Okay, okay, sir.
You don't bother. Even if you didn't understand, whatever the important point I have said, that is enough for you. Okay.
Okay,
I am not limiting you, but I don't want to confuse you.
got it, sir. Yeah.
Yeah. So IgG is different, IgA is different, their functions are different. IgE is produced in allergic conditions. Immunoglobulin E, IgE is produced in allergic conditions. Okay. And IgA, which is called a secretory, IgA, it is present in the gastrointestinal tract, in the mucosal lining. Okay. The B cells, which are present in the intestinal tract lining, there they produce immunoglobulin A. It is also called as a secretory antibody. So it fights against all the antigens that are entering to the gastrointestinal tract, right? Did you understand?
Yes, sir. Yes, sir. Clear. Pretty clear. Sir, you are on I guess on guys, is it just me or sir is on hang?